Welcome back to the Deep Dive, where we unpack complex topics to give you those essential nuggets of knowledge. Today, we're putting on our detective hats and embarking on a deep dive into some of the most common and, frankly, most cunning bacteria you'll encounter in medicine, the Enterobacteriaceae. Think of it as building your first pathogen playbook.
Precisely. This family of microbes is an absolute master of adaptation and infection. I often call them the Swiss Army knife of bacteria because of their versatility, capable of causing everything from a mild stomachache to life-threatening sepsis. Our mission today is to equip you, our dedicated learner, with the foundational knowledge to understand their tactics, retain the key differences, and recall exactly why these distinctions matter in the clinic.
That's right. We're not just going to list facts. We're going to piece together a puzzle. We'll break down their general characteristics, identify their preferred battlegrounds in the body, and spotlight the unique strategies of some of the biggest players in this family. We'll use memorable anchors, acronyms, metaphors, make this material truly stick. And we'll even pose a few questions to get your diagnostic gears turning. Okay, let's start uncovering the clues. So when we talk about Enterobacteriaceae, what exactly are we up against? What's the common profile for this diverse family?
Well, at their core, these are a large and incredibly diverse group of gram-negative rods, bacilli. We're talking over 50 genera and more than 150 species. But here's the crucial insight, the kicker for you. Less than 20 of these species are actually responsible for over 95% of the infections you'll see clinically.
Wow, only 20 doing most of the damage.
Pretty much. This family causes over 70% of urinary tract infections, 30–35% of septicemias, and many intestinal infections. They are truly ubiquitous.
Ubiquitous indeed. And their origins are as varied as their infections, right? Some come from animals.
Exactly. Some, like most Salmonella species or Yersinia, come from an animal reservoir. Others, like Salmonella typhi or Shigella species, have human carriers.
And then there are the resident troublemakers, organisms like Escherichia coli that already live inside us, just waiting for an opportunity to cause disease. Endogenous spread, we call it.
Right, the inside job.
Precisely. Now let's talk about their physiology, the fundamental traits that serve as their basic ID card, you could say. You can use the acronym FERN to remember these key characteristics, which are often the first clues we get in the lab. Facultative anaerobes.
Meaning they can live with or without oxygen. Adaptable.
Very adaptable. Gives them a huge advantage in various body environments. Easy growth. They grow rapidly. Simple nutritional needs. They're not picky eaters, making them easy to culture.
Okay.
Reduced nitrate to nitrite. A key biochemical reaction, kind of like a quick fingerprint in the lab, helps distinguish them. And negative oxidase. Unlike many other gram-negative rods, they lack the cytochrome oxidase enzyme. This is a crucial differentiator for lab identification. And it's worth adding, they also ferment glucose and are catalase positive. Plus, you mentioned many pathogenic members typically do not ferment lactose, and they resist bile salts. More clues.
Important clues, definitely.
So when you're first thinking about a patient, how quickly can these general traits actually help you? Almost like a first filter, even before you get specific lab results back.
Oh, surprisingly quickly. I mean, knowing, for instance, that Enterobacteriaceae are gram-negative and oxidase-negative immediately rules out a huge number of other bacteria that might cause similar symptoms. That FERN profile, combined with the site of infection, where the problem is, very quickly narrows your differential diagnosis down to this family. It's a powerful initial sorting mechanism, really essential for guiding early treatment decisions.
That's a great insight. So these basic characteristics really guide our thinking from the get-go. Now, within this big family, you mentioned different types of troublemakers, like an invading army versus sleeper agents.
That's a powerful analogy, yes. The invading army are what we call primary pathogens. These are the species that almost universally cause disease when they encounter a susceptible host. We're talking species like Salmonella enterica, Shigella, Yersinia pestis, Yersinia enterocolitica. They don't need an invitation to cause trouble. They're built for it.
Always looking for a fight. And then the sleeper agents, the opportunistic pathogens.
Exactly. These are often part of our normal flora, quietly living their lives until a weakness or an opportunity arises. Maybe the host's immune system is down, or they move to a part of the body where they just don't belong.
Like an E. coli leaving the gut to cause a UTI.
Precisely. Common opportunistic culprits include many strains of Escherichia coli, though some E. coli are primary pathogens, it gets tricky, Klebsiella pneumonia, Proteus mirabilis, Enterobacter, Escherichia. The list goes on. The key insight here is that knowing if a pathogen is primary or opportunistic tells us a lot about the host's vulnerability. It's the difference between an uninvited guest breaking down the door and a trusted resident turning rogue. Understanding that initial intent is often the first critical diagnostic clue.
That's a brilliant way to put it. Really clarifies how understanding their nature informs our initial assessment. But how do these bacteria actually cause disease? They must have some serious weaponry, right?
Whoa, they absolutely do. They're armed with a sophisticated, let's call it a special forces toolkit of virulence factors. These are the specific weapons and strategies they use to evade our defenses, attach to our cells, and, well, cause damage.
This arsenal is fascinating, almost like a specialized military unit.
Yeah.
You mentioned the type 3 secretion system before injecting proteins directly into host cells. That sounds like, wow, a masterclass in cellular manipulation. How does that directly translate into the disease we see? And what are some of the other key weapons? We can use the acronym FLOCKS, right?
Yes, FLOCKS is a good way to remember some major ones. F is for flagellar antigen, the H antigen. These are like tiny propellers giving bacteria mobility. Lets them move through our bodies, spread the infection.
Got it. Mobility.
L is for LPS, lipopolysaccharide,
Yep.
a major component of their outer membrane. It includes the O antigen, which helps them sort of hide from immune detection, and lipid A. Lipid A is the real chiropower, the endotoxin activity. Triggers a strong, sometimes overwhelming inflammatory response.
The fever and shock component.
Exactly. Then C is for capsular antigen, the K or Vi antigen. This acts like an invisibility cloak surrounding the bacteria, helping them resist phagocytosis by our immune cells. It's a key defense for them.
Clever, like stealth tech.
Sort of, yeah. K stands for key secretion systems, especially the type 3 secretion system, which, as you said, is like a molecular syringe. It injects bacterial proteins, effectors directly into our cells, manipulates them to the pathogen's advantage, changing cell function, disrupting barriers, maybe promoting uptake by the cell.
Wow, that's direct sabotage.
It really is. And finally, S is for cytophores. These are like metal detectors or scavengers. They grab essential iron from the host. Bacteria desperately need iron to grow and multiply, so they literally steal ours.
They steal our iron. Okay, FLOCKS, flagella, LPS, capsule, key secretion system cytophores. That's quite an arsenal.
And we can't forget antimicrobial resistance. It's an increasingly critical virulence factor, often encoded on plasmids or other mobile genetic elements. It directly impacts our ability to treat these infections effectively, making the bacteria not just skilled attackers, but incredibly resilient too. It's an evolving challenge for us.
A huge challenge. Okay, with that special forces toolkit understood, let's deploy it in our minds as we meet the specific masterminds, starting with the notorious Salmonella. What makes this particular genus such a dual threat, causing both, mild inconvenience and potentially deadly systemic disease?
Salmonella is indeed fascinating because of that dual nature. Most clinically relevant species belong to Salmonella enterica, but within that, there are numerous serovars differentiated by their OH and sometimes K antigens, those things we just talked about. You'll often see them referred to by their serovar name like S. enterica serotype typhi, often shortened to S. serotyphi. These distinctions are absolutely critical.
And as you said, Salmonella enterica primarily causes two very distinct types of diseases: gastroenteritis, the more common one, and the much more serious enteric fever, typhoid fever. Let's start with the common one, gastroenteritis or non-typhoidal salmonellosis. This is often caused by serovars like S. enteritidis and S. typhimurium, right?
That's correct. Transmission is typically through ingesting contaminated food, poultry, dairy, eggs are common culprits, or contaminated water. Also the fecal-oral route. Symptoms usually appear pretty quickly within, say, 6 to 48 hours. Nausea, vomiting, diarrhea, fever, abdominal cramps. The usual suspects for food poisoning.
And what's happening inside the gut? How does it cause this?
Well, S. enterica attaches to the lining of the small intestine. It specifically invades the M cells in Peyer's patches. Those are specialized immune surveillance spots, and also regular gut lining cells, the enterocytes. Once inside, they replicate within these little membrane bubbles, endocytic vesicles. They use their type 3 secretion system, that molecular syringe, to inject proteins. These proteins help them attach, get engulfed, and then multiply inside. This leads to local inflammation, production of enterotoxins, and that results in the classic symptoms. Sometimes it can even get into the bloodstream and cause septicemia, but usually it stays fairly local.
Diagnosis is usually a stool sample.
Usually, yes. And for treatment, rehydration is absolutely key. Fluids, electrolytes. And here's a critical point for you as a clinician. Antibiotics are generally not needed for typical Salmonella gastroenteritis. It's often self-limiting. In fact, antibiotics can sometimes prolong how long someone carries the bacteria.
Good to know. Avoid antibiotics usually. But it's a huge global burden, you mentioned.
Huge. Estimated 93.8 million cases and 155,000 deaths annually worldwide. Staggering numbers.
Truly staggering. Now let's talk about its insidious cousin, enteric fever, or typhoid fever. Much more serious, life-threatening, caused by S. typhi and S. paratifi.
Yes, a completely different beast, really. Transmission is, again, through contaminated food or water, fecal-oral route, or contact with asymptomatic carriers. But the incubation period is longer, typically 10 to 14 days. And the symptoms are different. High fever, headache, often constipation before diarrhea, abdominal pain, general malaise. And it can persist for 3-4 weeks if untreated.
And the pathogenesis, that's where it really differs, isn't it? It doesn't just stay in the gut.
Not at all. This is the key. After ingestion, S. typhi invades the small intestine but then rapidly enters the bloodstream. Systemic invasion. From there, white blood cells actually carry the bacteria to organs like the liver, spleen, and bone marrow. And there, they multiply. They set up shop, then they re-enter the bloodstream in higher numbers and invade the gallbladder, the biliary system, and lymphatic tissue of the bowel. Multiply again to high numbers before finally being shed in the stool.
Wow, that's a complex journey. And there are stages to this illness. You mentioned fast.
Yes, untreated, typhoid fever typically progresses through 4 stages. Fast helps remember them. F is for fever, typically stepwise, getting higher each day. Also, relative bradycardia, a slow heart rate despite the high fever, which is unusual. Malaise, headache, cough, maybe nosebleeds.
Okay, stage 1.
A is for abdominal distension and pain. Continued high fever, weight loss. And the characteristic rose spots on the skin, faint pink spots usually on the trunk. You might also see an enlarged liver and spleen, hepatosplenomegaly. Delirium often starts or worsens here.
Getting serious.
S is for severe complications. This is where things like intestinal hemorrhage or perforation can happen. Encephalitis, severe dehydration. The delirium gets worse. Very dangerous stage.
And T?
T is the turning point. Defervescence, where the fever gradually starts to fall, hopefully as the body starts to win, or if complications overwhelm the patient.
Complications like overwhelming infections, pneumonia, kidney failure, peritonitis, these can be fatal.
Tragically, yes. Diagnosis relies on blood cultures early on, later stool, bone marrow, or urine samples. PCR helps too. And unlike the gastroenteritis, treatment requires antibiotics. Fluoroquinolones are often used, though resistance is a growing issue.
And the carrier state, Typhoid Mary, that's real.
Oh, very real. About 3–5% of infected individuals become asymptomatic carriers. Their infected gallbladders often act as a reservoir, continuously shedding bacteria in their feces, sometimes for years, without any symptoms themselves. Silent spreaders.
So back to our earlier question, what makes typhoid fever so much more insidious and deadly than the typical Salmonella gastroenteritis, even from the same genus? What's the core insight?
The core insight is really in that invasion strategy and systemic survival. While non-typhoidal Salmonella mostly causes local damage in the gut, leading to that acute, often self-limiting diarrhea, S. typhi is a master of systemic evasion. It uses its virulence factors, particularly its capsular V and antigen, to essentially hide from immune cells, get into the bloodstream, and then multiply within immune cells in places like the liver, spleen, bone marrow. This ability to travel, replicate throughout the body, and then establish that chronic carriage state in the gallbladder. That's what makes it so much more dangerous and insidious. It's the difference between a localized skirmish and a full-scale stealthy invasion.
Yeah, systemic invasion makes all the difference.
Okay, next up we have Shigella, the cause of shigellosis or bacillary dysentery. How does this one compare in its attack strategy? Shigella is another intestinal invader, but with its own distinct and quite brutal approach. The clinically relevant species are grouped into 4 serogroups: S. dysenteria, S. flexneri, S. boydii, and S. suni. Unlike Salmonella, these are non-motile, they don't have flagella, and they're non-capsulated. Importantly, humans are their only known host.
Only humans. That simplifies things a bit, maybe. But transmission is notoriously easy, right? Very low infectious dose.
Extremely easy. Primarily person to person, but also contaminated food, water, surfaces. And yes, a tiny number of organisms, sometimes just 10 to 100 bacteria, can cause disease. Highly transmissible, especially in places with poor sanitation.
Symptoms appear quickly, 1 to 3 days, And the key is bloody stools.
Yes, typically 1 to 3 days incubation lasts 5 to 7 days. Abdominal cramps, fever, diarrhea, and critically bloody stools. That's the hallmark of dysentery, caused by the damage to the intestinal lining.
And how does it cause that damage? What's its invasion tactic?
Well, Shigella also proliferates rapidly in the small intestine, then attaches to M cells in the Peyer's patches and also the colon lining. It invades these epithelial cells, but instead of staying in vesicles, it escapes into the cell's cytoplasm and replicates there. And here's the clever bit. They use the host cell's own actin filaments, part of the cell's skeleton, to build little tails, like rocket boosters, propelling themselves directly into adjacent cells.
Wow, cell to cell spread without going outside?
Exactly, spreading the infection laterally through the tissue, avoiding exposure to the immune system outside the cells. Very efficient, very damaging.
And the Shiga toxin, that plays a big role.
A major role, particularly for S. dysenteria. The Shiga toxin is a potent exotoxin that directly kills host cells by inhibiting protein synthesis. This contributes massively to the severe inflammation, the extensive cell damage, the ulceration of the mucosa, and that characteristic mucus and blood you see in the stool.
Yeah.
But here's a key distinction from S. typhi. Shigella is usually quickly contained if it does get into the bloodstream. It's phagocytosed and killed. It generally doesn't penetrate beyond the intestinal lining. It causes intense, localized damage, but usually not systemic spread like typhoid.
So very nasty locally, but doesn't typically go systemic. Diagnosis is stool sample again.
Yes, stool sample. Treatment primarily focuses on rehydration, of course.
Yeah.
But unlike typical Salmonella gastroenteritis, antibiotics like ciprofloxacin are often recommended for shigellosis, especially severe or prolonged cases. Shigella generally responds well to antibiotics.
Another key treatment difference then. This distinction in systemic versus local infection is really profound, isn't it?
It absolutely is. The contrast is stark. Shigella unleashes this furious localized assault, causing severe damage, but it largely stays contained within the gut lining. Salmonella typhi, as we discussed, opts for that stealthier systemic invasion, which, while may be slow to cause the initial gut symptoms, can ultimately be far more devastating systemically if it's not checked. Understanding these different invasion strategies is such a powerful lens for interpreting clinical presentations.
Okay, moving on to a name everyone recognizes. Escherichia coli, or E. coli. It's everywhere, right? Part of our normal gut flora for the most part. But it can also be a formidable villain. What makes E. coli so versatile? Why does it wear so many different hats, pathogenically speaking?
Indeed. E. coli is usually a harmless, even beneficial inhabitant of the human intestine, commensal. But its versatility comes from its genetic flexibility. It can easily acquire new genes, often via plasmids or bacteriophages, that encode virulence factors. So certain strains become highly pathogenic, causing everything from gastroenteritis to UTIs, neonatal meningitis, sepsis, a huge range. Their key virulence factors include a wide array of adhesins, allowing them to stick to different tissues and various exotoxins. This broad range of capabilities is precisely why it's so important to identify the specific strain or pathotype.
Transmission is usually fecal-oral. Contaminated food, water, undercooked beef, unpasteurized milk.
That's typical, yes. But as you said, not all pathogenic E. coli are created equal. Different strains or pathotypes cause different diseases using different mechanisms. We can categorize the major gastroenteritis-causing strains with the acronym MEATS, each with its own signature attack style.
Okay, let's break down MEATS. E is for EHEC.
Right, EHEC, enterotoxigenic E. coli. This is the classic cause of traveler's diarrhea and also a major cause of infant diarrhea in developing countries. Its main weapon is toxin production. These strains produce heat labile, LT, and/or heat-stable, SDA, enterotoxins. These toxins mess with the fluid transport in the small intestine cells, essentially making them secrete lots of water and electrolytes. Result: profuse, watery, non-bloody diarrhea, like turning on a faucet in the gut.
Traveler's toxin. Got it. Next E in MEATS.
EHEC, enteropathogenic E. coli. This one also primarily causes infant diarrhea, particularly in low-income countries. It uses adherence factors. It attaches very intimately to intestinal cells using things like bundle-forming pulli and a protein called intimin. This causes physical damage. Effacing the microvilli, the tiny absorptive fingers on the cells, impairs absorption, causes watery diarrhea. It literally bulldozes the absorptive surface.
Okay, EHEC damages the structure. Then EAEC.
EEEC, enteroaggregative E. coli. You see this causing persistent watery diarrhea, sometimes lasting weeks, vomiting, dehydration too, especially in infants, people with HIV, and travelers. It uses special fimbriae to aggregate or clump together, forming thick biofilms on the intestinal surface, like a sticky carpet. It also produces various toxins. This aggregation likely impairs function and causes inflammation.
Sticky carpet. Okay, the S in meats. This is the scary one.
This is often the most dangerous one, yes.
Yeah.
EHEC, Shiga toxin-producing E. coli, also known as EAEC, enterohemorrhagic E. coli. These cause watery diarrhea that progresses to grossly bloody diarrhea hemorrhagic colitis. And they carry a high risk of a severe complication called Haemolytic Uremic Syndrome, US, which involves kidney failure, low platelets, and hemolytic anemia. The infamous E. coli O157 H7 strain is the prime example. Their main weapon is producing potent Shiga toxins, STX1, TS2, similar to the one from Shigella dysentery. These toxins damage blood vessels, especially in the gut and kidneys.
So bloody diarrhea plus risk of kidney failure. That's serious. And the last E in meats.
EIEC, enteroinvasive E. coli. This one is less common in developed countries, but it causes fever, cramping, and watery diarrhea that can progress to dysentery with scant bloody stools. These strains behave much like Shigella. Their key feature is invasion. They penetrate and replicate within intestinal epithelial cells, causing inflammation and cell destruction.
So EHEC, toxin, EHEC pathology adherence, EAEC equals aggregation. STESAHEC, Shiga, toxin hemorrhage, HUIS, EIEEC, invasion. That meets acronym really helps differentiate them.
It does. And for E. coli O157 often found in things like undercooked ground beef or unpasteurized milk or contaminated produce, a very low dose can cause disease. Diagnosis is by stool sample, often needing specific tests for the Shiga toxins or the O157 serotype. Treatment for stec infections like this is primarily supportive, aggressive rehydration, and electrolyte correction, managing potential complications like HUS. And here's that critical point again, worth repeating. Antibiotics are generally contraindicated in STESAHEC infections. There's evidence they can actually increase the release of Shiga toxins as the bacteria die, potentially increasing the risk or severity of HUS.
Wow. So for this specific dangerous E. coli, antibiotics might make things worse. That's a huge clinical takeaway. But for other E. coli infections, like UTIs, meningitis, sepsis caused by other strains.
Oh, absolutely. For those infections, prompt antibiotic therapy is essential. It really highlights why identifying the specific type of E. coli infection is so crucial.
Absolute critical distinction. Okay, our final deep dive pathogen for today is Yersinia. What's its unique claim to fame? What makes it stand out?
Well, the Yersinia genus includes Y. enterocolitica and Y. pseudotuberculosis, which cause gastroenteritis or enterocolitis, sometimes called yersiniosis, and also the famous one Y. pestis, which causes plague, but we're focusing on the enteric ones today.
Right, and what's really fascinating about the enteric Yersinia species is their ability to grow at low temperatures, right? Even in the fridge.
Exactly. That's a key feature. They can propagate even in refrigerated and vacuum-packed foods. This makes them a stealthy foodborne threat, particularly with things like pork, dairy, meat, produce. Transmission is primarily through ingesting these contaminated foods. Less commonly, but with high mortality, it can be transmitted via contaminated blood transfusions because it can grow in stored blood.
Scary thought. Symptoms: diarrhea, fever, abdominal pain. How long does it take?
Yes, those are the main symptoms. Usually appear within 4 to 6 days after exposure and can last quite a while, sometimes up to 2 weeks, or even become chronic. It's seen more often in cooler climates, Europe, North America.
And there's a specific clinical sign, isn't there? Mimicking appendicitis.
Yes, that's a really key clinical manifestation. The abdominal pain can be severe and localized to the lower right quadrant, the area of the terminal ileum. This is often due to inflammation and enlargement of the nearby mesenteric lymph nodes. It can very closely mimic acute appendicitis. We call it pseudoappendicitis.
That's a powerful diagnostic pitfall, isn't it? Imagine a clinician in the ER faced with those symptoms, right lower quadrant pain, fever, making that crucial differential diagnosis between appendicitis needing surgery and yersiniosis that usually doesn't.
Precisely. It really underscores why understanding these nuances, keeping these less common but important pathogens in mind, is so critical. It directly impacts the patient's journey, potentially saving them from an unnecessary operation. Diagnosis is typically through a stool culture, though Yersinia can be a bit tricky to grow, sometimes needing special cold enrichment. Treatment, often self-limiting, so antibiotics aren't always needed for simple gastroenteritis. But for more severe or systemic cases, or if the patient is immunocompromised, antibiotics like cephalosporins might be prescribed.
Okay, so we've covered a lot today. From the broad characteristics of the whole Enterobacteriaceae family, that fern profile, to the specific tactics of Escherichia virulence factors, and then the distinct strategies of Salmonella, Shigella, the many faces of E. coli, and Yersinia.
We have. We've seen how their origins, their basic physiology, and especially their specific virulence factors create a unique profile for each major player. And understanding these nuances, whether a pathogen is primary or opportunistic, its specific weapons, how it's transmitted, the exact disease it causes, whether it stays local or goes systemic, is absolutely critical for you when you're facing a patient in clinical practice.
Which brings us back to our final question for today. We've seen how diverse the symptoms are, how different the underlying mechanisms can be, and crucially, how treatments can vary effective for one, potentially harmful for another, like with Escherichia coli. So why is it so fundamentally critical to identify this specific pathogen, especially when you're thinking about treatment strategies? What's the ultimate takeaway for patient care here?
The ultimate takeaway is precision. It's about targeted therapy. Knowing the specific pathogen is the difference between, well, blindly treating symptoms and targeting the actual cause of the disease with accuracy. Think about that E. coli O157.H7 example again. Giving antibiotics, which might seem like the standard thing to do for a bacterial infection, could actually worsen the outcome, potentially triggering HUS. Conversely, not giving antibiotics for typhoid fever or E. coli sepsis would be disastrous. It's all about patient safety and effective care. You have to match the right countermeasure to the right microbial threat. Without precise identification, you're essentially fighting in the dark, and that's not good medicine.
Fighting in the dark. That sums it up perfectly. Okay, and that wraps up our deep dive into the complex, sometimes dangerous, but always fascinating world of Enterobacteriaceae. We've untangled their family tree, explored their sneaky survival skills, and delved into the distinct pathologies of some of their most infamous members.
We really hope that the metaphors, the acronyms FERN, FLOX, FAST, MEATS, and the questions we've posed have given you a solid, memorable framework for understanding these crucial pathogens. Remember, this knowledge isn't just academic. It's a vital part of your future medical toolkit. It's what helps you make informed, sometimes life-saving decisions at the bedside.
So, what does this all mean for you, the listener? It means when you encounter a patient with gastrointestinal distress or a UTI or maybe an unexplained fever, you'll have a systematic way to think about the potential culprits. You can start filtering using those clues we talked about to narrow down the possibilities and guide your investigation towards the best course of action. Keep exploring those reliable medical sources, keep asking questions, and remember, every piece of knowledge you gain now will make you a more effective and compassionate healer down the line.
Indeed. And maybe a final thought to leave you with. While we focus mainly on the acute presentations today, the immediate illnesses these bacteria cause. What are the long-term public health implications? Think about chronic carriers like with typhoid. Think about the sheer volume of infections globally. And critically, think about the escalating crisis of antimicrobial resistance within this family. How does that change the game, not just for individual patients, but for global health strategy in the coming decades?
That's a powerful thought to end on. The ripple effects go far beyond the single infection. Thank you for joining us on this deep dive, everyone, and keep learning.